this is me. My name is Dr. Takis Athanasopoulas. I'm a university lecturer in a local university here, University of Wolverhampton. And also I'm a research consultant at the University of London and for various companies. And we were performing some design vector optimization studies as part of a consortium of scientists that is called the Collaboration for AIDS Vaccine Discovery. And I will talk to you a little bit about that and what we are doing in the field of HIV vaccines. So as you recognize this paper, François Barré-Sinouse and Luc Montanet, they got their Nobel Prize back in 2008. But the quest to find a cure against HIV/AIDS is long-standing with more than 45 years these days. And the AIDS epidemiology is all over, widespread all over the world, but the most critical regions is in the area of sub-Saharan Africa. And this is because of multiple diversity constraints in the origin of HIV. Normally because of errors in the reverse transcriptase and highly genetic recombination, but also in other factors like oxidative stress and the ApoBec3 factors. So based on the structure of HIV and their classification, there are lots of targets in the GAG gene pool and the envelope in the reverse transcriptase of the virus. And lots of research has been conducted to identify potential targets. Our part was to try to assist a multi-consortium of partners for an HIV vaccine. The attempts were not successful, otherwise you would be known in the news, but I can tell you some progress with this. And of course, the virus tropism and replication cycle, you are all aware of this, so I will not expand, but the CD4 and some core receptors like the CCR5 and the CXR4 are essential parameters in the entry of the virus. So conventional approaches towards HIV control with a highly antiretroviral medication are quite successful to control the epidemic, but it requires a long life commitment. Not all the patients can receive highly antiretroviral target therapy, is around 30%. And for this reason, the scientific community was put forward some ideas for a genetic vaccine. But in order to control therapeutically the virus, lots of elements and lots of commitment has to come together. So big international partnerships, starting from basic research, clinical research, public health organizations, private sector, and a multi-million components. New targets against HIV are identified almost daily. And you may have heard terms like short hairpin RNA inhibition or microRNAs or the new CRISPR system that is quite fashionable and directed against the co-receptor, the CCR5, or even microbiocides. And as part of this regime, we have developed a kind of prophylactic therapeutic vaccination approaches. So this path for discovery has to acknowledge the genetic variability of the virus and the viral evasion of the immune system, and also to try to early establish and counteract the viral reservoirs that is in the mucosal area. So in order to do this, lots of approaches have been tested. Back in 7, 8, a Merck vaccine that is called the STEP trial was introduced, but that was unsuccessful. They introduced a prime boost component using the gag-pol envelope components of the virus. But it was an unsuccessful vaccination, and actually it was thought that lots of the design was erratic, because the patients that they received actually the vaccine, they were more promiscuous to HIV infection rather than they were counteracting the infection. As part of a huge network of scientists, the Collaboration for AIDS Vaccine Discovery, funded by this guy, this couple here, Bill and Melinda Gates Foundation, and of course supported by multimillionaires like Buffett and others. We were lucky to receive some funding as of 2006. Back in 2006, we were one of the first consortia that we received funding from the foundation. Then, at the origins, back in July 2006, there were 19 consortia, and they have received up to then 327 million pounds. So it's a huge investment of the scientific community towards accelerating the path for the vaccine. And of course, they had different goals: to accelerate HIV vaccine research, conducting collaborative research, to design novel vaccine candidates, and introducing the most promising candidates to preclinical and clinical trials. And of course, there was a huge consortium talk, crosstalk. So we were part of only one of these consortia. That was namely the Stephen Patterson consortia. So I was part of this consortium. We were around 20 scientists, and imagine there were multiplicities of 20 for each of the other consortia, so a huge amount of effort and scientists. Some of the coordinators you can see here: Bob Gallo in the Maryland, Haynes, Ledgevin, Thomas Lerner, Ho, Juliana McElrath, Bruce Walker, Leo Stamatatos, another fellow Greek, Steve Self, and Greenberg. So there are lots of people that they were coordinators of this original consortia, 19. Nowadays, just last year, they were from 19, they jumped to 37 consortia. So we have 37 vaccine consortia only from the Collaboration for AIDS Vaccine Discovery. We are not going to mention the HTN or other huge consortia for HIV. So a huge effort, and each consortium was dedicated to different variations like immunogen design, passive immunization, replicating vectors, protective antibodies or port of entry, and everything was coordinated by a central services facility that was bringing all the information like a cloud together, was taking the data. They had the mouse immunology vector core, and they could help you out for all your immunological experiments. The part of our consortium was to develop a candidate HIV-1 clade C vaccine that could evade in theory, the host anti-vector immunity and stimulate broader persistent CD8 cytotoxic T cell responses. And this was using, again, an adenovirus, but we had different serotypes. So we started with adenovirus 5, that is the common serotype, but also we expanded in adenovirus 11. And I will tell you a little bit what is the significance of different serotypes as we go along with the talk. So we were awarded around 9.2 million over 5 years, was spread the cost, and there were lots of collaborating institutions. And I was based at this institution in London, Royal Holloway University of London, for the particulates of research. Part of delivering an HIV vaccine has to do with tropical sub-Saharan Africa, where the temperatures of a successful vaccine can go up to 40 or 50 degrees. In order to circumvent this cold chain reaction, not to use freezers, because adenovirus is not viable for a long time after an hour or two. Let's say in a normal environment, you can do the experiment, but after a day, it will not survive outside of the -80 freezer. For this reason, we tried to formulate adenovirus microneedle skin patches in order to show some thermostability, to indicate some thermostability. So this is about adenoviruses. I'm sure you have heard before, double-stranded DNA viruses. The adenovirus 5 is the common cold flu virus and causes benign respiratory tract, upper respiratory tract infections. We wanted to introduce this virus into dendritic cells, and the dendritic cells of the skin can migrate and go to mucosal surfaces, so you can induce mucosal immunity by introducing it to the skin. And the technology was a really simple idea, so you had some microneedle array technologies, we call them. They were looking like skin patches, like the Hansaplast that you put on the hand, and inside the patch, that was made by a component called carboxyl methyl cellulose. That was the key component of the patch. You could put the adenovirus, but in order for the adenovirus to be thermostable, you had to coat it with different layers of sugars. And this is what we did. We did different experiments with sucrose, raffinose. We tested around 10 sugars. And from all the sugars, we found a combination that could outlast the virus thermostability up to 3 months. And here are more experiments. And here, for example, for a virus that expresses GFP, you could show that it can be thermostable for 40 degrees for a month in a sucrose storage plus some other solutions that we had available. So that was good news. You could maintain the virus. You can air dry. Sucrose trehalose are preferable preservatives. And then the next step was to do the actual vaccine approach. In order to have some immunogenicity, to expand in broader immunogenicity for the virus, we followed up 2 strategies. One was called ubiquitination. We adopted artificial ubiquitins on the beginning, the 5' end of the molecules, in order to target more efficiently into the proteasome. And the second one was the gag fragmentation. So if we wanted to vaccinate against the gag, we compartmentalized, we fragmented the gag into multiple fragments, and each of these fragments were to generate a virus, and this virus was to go into the skin in order to broaden the immune response repertoire. Lots of work, because imagine that if you do this for every single component of the virus, and I will show you a little bit, we made only 10 fragments for the gag part of the HIV. That means 10 viral vectors for the gag. And this is the HIV component that can go, in theory, into patients. But also, you have to test it in non-human primates, so you have to do the same exactly for the SIV component. So really lots of work and a multi-step procedure. So we followed this and different serotypes. So we did serotype 5 and serotype 11 for this procedures. So here you see as an example the full gag and the analysis of the full gag and how the full CN54 gag gene was mono or tetra ubiquitinated. So this is the ubiquitination approach. And we followed up this process. We did experiments in mice. It worked well, but the end point result was not so promising. So the ubiquitination approach apparently didn't offer much. And you can see lots of experiments, fragmentation, how the fragments of 0 and mono ubiquitin are appearing on Western blots, and so on and so forth. The second approach, though, was more promising. So if you fragment a gene, and the fragments, they correspond to individual epitopes, and these are overlapping epitopes of the same gene, then you can broaden the repertoire of the immune response. So that was quite encouraging. So we followed an epitope prediction model, map. I'm not going to bore you into details. And from this epitope prediction map, we identified how to concatenarize the gag gene of the vector into fragmented 10 fragments. And this was was analyzed. We were synthesized by a company called GeneArt that was in Germany, but now is a part of Life Science Technologies about codon optimization of the individual microfragments. And you can see here the molecular size of each microfragment, the corresponding. Some were 0 ubiquitinated, some were mono, and some were tetraubiquitinated. So you had a combination of 30 fragments that went into 30 adenovirus vectors. And here you can see how they look, let's say some of the fragments when you analyze them on a Western blot. And you can see that the tetraubiquitinated fragments appear to cover the full repertoire. So you have 0, mono, D3, and tetraubiquitinated approaches on the Western blot. So everything was good. We did massive production of adenoviruses in the lab. So we did it in-house, but also we used a company in America called Capital Biosciences. We generated stocks of HIV, we generated stocks of SIV fragments, and then we went into non-human primate, and we did some experiments in non-human primates. But you can read more in this publication. So we published back in 2009 this paper in PNAS that adenovirus vector vaccination can induce expansion of memory CD4 T cells using mucosal homing phenotype. So that wasn't very good news for the STEP trial, but was good, let's say, as an approach, as an understanding of the HIV vaccination field. Another publication, again, PNAS, Langer-negative dendritic cells can promote potent CD8 positive T cell priming by skin delivery of adenovirus microneedle arrays. So that was an interesting study that was conducted. And then most recent, last year, that the ubiquitin doesn't work. So that was a negative study. And a fusion of ubiquitin to HIV GAG impairs the human monocyte-derived dendritic cell maturation, and as a result, reduces the ability to induce GAG T cell response. So this is bad news. So ubiquitin, while you expect it to go and target into the proteasome, it doesn't work as anticipated. So perhaps don't try it for your vaccination approach. In my lab now, I have converted. I'm not really an immunologist per se. I'm a gene therapist. So we are working using adenovirus, but also adeno-associated virus or lentivirus to cure genetic disease. Not to in a sense, we want the opposite effect, to minimize the immune response and to cure the disease. But sometimes it's useful to know. And we were working with lenti-based systems and adeno-associated virus systems. And recently, in the literature, you can use these system approaches to induce vaccination, because you can use it as immunoadhesin molecules, as diffusion molecules. So you can use, for example, muscle as a platform, and you can express antibodies, monoclonal antibodies or polyclonal, against the recipient. So it's quite interesting approaches. Following the adenovirus strategy, in just in a couple of minutes I will finish the talk, we followed, not as a part of the collaboration for AIDS vaccine discovery, but as part of the Blue Horizons, is the same program for Gates Foundation, but they give you small amounts. They give you around $100,000. And if you prove that your study is reasonable as a proof of concept, they can give you millions afterwards. So we were lucky, and actually it's highly recommended, because you can write only 2 pages A4, and you can get $100,000. So if you have a clever idea, you can go and apply to GCE programs. As part of one successful program, we wrote with Professor Dixon a lentivirus vaccination strategy for HIV. And that had to do, as an approach, a multi-epitope virus-like particle envelope vaccine development. It's as simple as a concept, as an idea. So you know the HIV virus, that is complex in structure, but the VLPs are simpler, are self-assembling, non-pathogenic, non-replicating particles, and genome-less. They don't have any RNA, they don't need the reverse transcriptase mechanism. But our approach was to generate iterate library. So each library of the envelope of the outside of the virion was slightly different. And you used millions of these slightly different epitopes. You construct them in plasmids, and afterwards they self-assemble and reassemble like virus-like particles. So you can make thousands of hybrid, mosaic, let's say, virus-like particles. Okay, what is the importance? Okay, so we did two design approaches, one with adenovirus, the fragmentation worked. Afterwards, we did this approach. We didn't go any further because we weren't lucky to get the 1 million funding. So this was frozen. But recently, you may have heard in the news from Harvard that a combination vaccine approach, essentially using an adenovirus serotype 26, that is similar actually to 11, vector vaccine to prime the immune system. And then if you boost, but not with the same adenovirus, but using a purified HIV envelope protein, was very successful in simian, in non-human primates. It showed actually 50% of the primates were protected against the simian virus infection. So this just in the news, July 2015. In Science, you can read all about it. And because it was successful, Johnson & Johnson initiated human clinical trials immediately after the publication of the study, just last month. So that shows if you use two conceptually different approaches, but let's say an adenovirus vaccination as a prime, and to come with an envelope protein afterwards, to use a VLP, particle approach, or a protein approach as a boost, then you can have some type of prophylaxis against the disease. Still, we haven't achieved lots. We haven't managed to generate an effective HIV vaccine, although there were successful efforts, and in Thailand, they were announced 3-4 years ago a regime that was protective to a 33% percentage of patients. But Bill Clinton in 1997 set up a 10-year goal. So now Bill Clinton is not a president anymore. Maybe Donald Trump, God help us all. And vaccine development remains the #1 priority of AIDS research, and perhaps the best hope against HIV infection, but still remains elusive. So I would like to thank you for your attention, and thank you for the opportunity to give me this panel for a couple of minutes. Hope it worth waiting. I know that you were eager to go have some lunch. These are all the institutions that helped in the collaboration vaccines study. And of course, I would like to thank more my ex-boss. Now I'm independent, but we are having a close collaboration, almost 20 years collaboration, Professor George Dixon at Royal Holloway. Thank you very much for your attention. Thank you, guys. Yes?
So this may be a completely naive question because I'm not in an HIV field. So
Neither am I anymore.
So for the STEP trial, I was under the impression that the increased T cells in the mucosa that was creating a reservoir for HIV was one of the reasons that we saw increased infection in the vaccinees. Is that correct?
I think it's correct. It's correct. They didn't comprehensively give an answer of what may have happened. Still, we're analyzing samples. As part of this analysis, we published this 2009 earlier study in PNAS that was showing one branch, one approach, let's say, why this may have failed. But this is far than conclusive. So what you say is absolutely correct.
So then following up from that, how will any vector or any vaccine approach that generates T cells
can work? You're right. I mean, that was in the early days of the field. That was the panacea that this may work. And for this reason, generations of scientists, including myself, they were trying to find a T-cell vaccine. Whether it will work or not is not yet established. However, saying that, the recent study from the Harvard guys, it shows that it can partially offer some protection. It's 50%. Half of the population still is better than nothing. The Thailand vaccine that was in a canary pox-based vaccination, again targeting T-cell, partially, they claimed 33% of the population. So it will not be perfect. It will not have 100% protection in the population, but may be helpful together with other approaches. For me, personally, I would think that Baltimore's approach and other science approach targeting the co-receptors, let's say the CCR5, may be more promising. Yeah, so if you try to block the co-receptor, then you will have an HIV floating reservoir, but it will not be infectious in the cells. And that was proven from natural inhabitants like the German patient. That's still another actual examples, 2 or 3 other examples that they arise in the literature. And still after 20 years, they are healthy and protected from HIV. So me personally, I would say that other approaches may be more promising. You are right that we have, as a field, to reconsider approaches. Yes?
I would like to ask about the species difference, because as far as I remember at her trial, it did work in chimpanzee. They were 100% protected.
Yes.
So the hypothesis about the overproduction of T-cell response wouldn't really be true for chimpanzees, but it didn't work in humans.
Yes.
So that's something that we probably need to find out understanding.
You're absolutely right, Spot. Yeah, differences in species, and of course, the animal models, we are limited from the animal models and to extrapolate the response to human clinical trials. Yes, but this is what the field has to go through. So you have to do experiments in mice, that the immune system is totally different. Afterwards, you have to go to non-human primates, including chimpanzees, that may be different, and then test the real thing into human and prey that it may work in a logical order. But it's not always the case. Thank you, guys. Did you have any lunch? Not yet? We are allowed lunch?